Hey guys, welcome to the very next video in this chapter. Now, in this lesson, I have a quick couple goals for you. First, I want you to be able to create your own functional DNA molecule with at least 36 bases and then turn it into a protein sequence. Yay, sounds fun, doesn't it? Basically, what you're going to do here is you're going to play God, because playing God is fun, and even if you're a mere mortal like the rest of us. What you're going to do here, guys, is pretty simple. You're going to have to use key terms like genetic code, codon, translation, anticodon, and gene expression to make your own DNA molecule and explain what the heck is going on here. So, first thing you have to be able to do is you need to be able to explain to me what might happen to a protein if the DNA was changed. Second, I want you to explain to me what the connection between proteins and genes are. And finally, I want you to explain the role to me of DNA polymerase in making proteins. So, first and foremost, the protein review. But I know many of you guys are probably thinking to yourself, what is a protein? Or what is amino acid? Well, proteins and amino acids we covered way back in chapter 2. As you know, they're enzymes, they can do a lot of things, they're very powerful, they're basically what make you you in lots of sense. Well, a protein is basically a whole bunch of amino acids that have been combined together. We call this combination a polypeptide. So if you see here, each one of these little orange, I guess orangish? They're not really orange, are they? They're kind of like, I don't know what the color that is, honestly. We'll call it reddish-orange. The reddish-orange little balls right here, each of these balls represents a different amino acid. And all these combined together equals a protein. So, now that that's out of the way, let's talk about translation. And no, I don't mean translation from English to Chinese. I mean translation in biology, which is when you turn messenger RNA into proteins. The general process for this is that the mRNA goes to the ribosome, the tRNA brings amino acids to the ribosome, and the ribosome makes the protein, which is your polypeptide, which is that fancy word I used from before. That's your basic process of translation. So, before I go into more detail, let's actually talk about, well, wait, how does that work? Well, there's a code, kind of like computer code. The code is that every 3 letters is going to equal 1 amino acid. So your combination of adenines, uracils, guanines, and cytosines is going to equal an amino acid. So for example, over here, if you look at this very, very complicated looking chart to your right, you're going to see like G, C, G. Well, what that means is if you see the combination GCG, or say the ribosome sees that combination, it's like, aha! That means I need to make the amino acid alanine. If it sees the combination U, 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 it knows it needs to make, oh, I have no idea how to pronounce that. We're going to call it phenylalanine. It knows you have to make the amino acid phenylalanine. So it uses these 3-letter combinations to figure out what goes where. Now, these 3 letters, the GCC, the AAA, all these letter combinations are called codons. So basically every 3 mRNA letters on an amino acid is considered to be a codon. So like GCA equals alanine. Now, there are some weird parts here. Now, it's not just that the ribosome grabs on the RNA and starts making proteins. That's not quite how it works. There is a start and a stop signal that basically says, now start making proteins, or now stop making proteins. Now for start, it's easy. It's always the combination AUG. So if you see AUG somewhere within your mRNA molecule, that's basically saying, all right, start. Now, once you have your start, you also have to have a stop. Otherwise, it's never going to stop making amino acids, and that's going to become a very, very big protein. So there are 3 different stop codes, and those stop codes you can see over here on the right. They're UGA, UAA, and UAG. So if the ribosome comes to any of these letters, it's like, all right, guys, time to stop. And that's how this whole process works. So you guys remember we had the normal process back here. We have the mRNA going to the ribosome, and it's going to make proteins. We've talked about here about how to read the code. Now we're going to go on to a little more details here. So the mRNA goes to the ribosome. And what happens next is the ribosome is going to read the codons. And the codons are those 3-letter combinations. It's then going to start making the protein once it finds the AUG. It will not start until it finds that AUG. If there's no AUG on the mRNA, it's not going to do anything with it. It's going to ignore it because it won't actually have a code to start. Then it's going to keep on making a protein, kind of like you see in this diagram over here to the right, where you're seeing this really, really long protein being made, and it's going to keep on making that protein until it reaches a stop codon. Once it reaches a stop codon, it's like, all right, guys, time to stop. Now, you might be wondering, what are these weird molecules over here on the right with a really weird shape that kind of makes them look like some weird chemistry device? Well, these right here are called tRNA. And tRNA, if you remember from the last lesson, basically bring amino acids to the ribosome that are going to attach to there. If you take AP Biology next year, you're going to learn to hate tRNA. It's a very annoying molecule, but don't worry, none of you guys are AP Biology yet. Now, this right here, as the bell's going off, so you can tell them at school still, is a video I'm going to show in class. But due to copyright issues, I don't want to get in trouble here on YouTube, so I'm not going to show you this video. You're going to have to download it off my website instead. Haha. Okay, so next, let's talk about anticodons. Now, there's codon and anticodon. Now, anticodon sounds kind of evil, but it's really not. The anticodon is basically going to be on the tRNA. So this right here, up here, is your tRNA molecule. The anticodon is the opposite of the codon. So what happens is that tRNA has 3 letters on it, just like the mRNA has 3 letters, and the tRNA is going to be searching for his codon. So basically think of tRNA as like, I must find codon-kun, and then all of a sudden it finds the codon here and saying, yay, I have found my codon friend. And then they say they're going to attach the amino acid. So basically, anticodon, opposite of a codon. It's found on tRNA. It's going to bind to the mRNA, and basically, the tRNA is going to look to the right codon to bind its anticodon to. And that's going to be kind of like the way the tRNA knows which amino acid to bring to the ribosome. So, now we're on the final part here, the central dogma of molecular biology. So, the central dogma is simple. It states that DNA is turned into mRNA, which turned into proteins. So what that basically means is that your genes are on the DNA is turned into RNA, and the RNA is turned into proteins. That's it. That's your central dogma of molecular biology. Sounds very complicated, but it's really not. Really, all this is showing you is how genes are expressed. It's telling you how genes are turned on and off, and how proteins are made. It's telling you why I have blue eyes and why other teachers have brown eyes. It tells you why I'm tall and people are short. It all comes down to is that my DNA is turned into RNA, which turned into proteins, and those proteins make me taller. So, my proteins are taller than your proteins. Thank you. Now, your alleles are just different proteins. So we go back to good old-fashioned Mendel. If we're talking about alleles, all we're really talking about is that there are different proteins. So the proteins between one allele versus another allele is just a difference in proteins. That's it. That's the only difference there. And that's it, guys. So, thanks for watching the video. I hope this makes some sense. If you have any questions, feel free to come talk to me in class, and I will do my best to try to answer them. Have an awesome day.